if it's even going to do that. Yep, it's just going to look weird. So we looked at our DNA secondary structure. What was that, Tuesday? Should be all kind of familiar with that. When we go through and look at our base pair alignments, so our adenine and thymine really only have 2 hydrogen bonding interactions. When we look at our cytosine and guanine, we end up with 3 hydrogen bonding interactions. So what you're primarily concerned about is what interactions are possible, and those are going to be what determines the base pairs. So when we talked about, what was it, Chablis's rule, that we had roughly equal amounts of adenine as we had thymine and equal amounts of cytosine as we had guanine, it was because of this double helix and that hydrogen bond pairing between those, and that it was very characteristic. A single adenine will only interact with thymine in a favorable interaction, whereas if it tried to bond to either cytosine or guanine, it doesn't allow for the proper alignment, and you end up disfiguring the double helix. Okay? You would for sure see a big disfiguration with the guanine. Why? If adenine tried to bond to guanine, what's going to be the issue there? What's the
hydrogen bonds?
We could push that, but somehow or another we'll be able to align. We'll probably set up some minimal interaction. In fact, we could probably see that nitrogen of our adenine. See if I can actually tag it on this. It'll let me do that. We could look at the nitrogen. Nope. Could look at the nitrogen of our adenine interacting with the hydrogen coming off of our guanine. And we could look at the lone pair on our carbonyl oxygen interacting with the hydrogen off of that guy. So we could still see a hydrogen bonding interaction.
When the hydrogen is the biggest problem would probably be during translation, you wouldn't get the protein.
What's the inner diameter of DNA? The inner diameter is 11 angstroms, which is exactly enough for 3 rings and a hydrogen bonding interaction. If we go through and put in adenine to guanine, do we have 3 rings? No, we've got 4. That's going to cause it to pucker and probably shift the whole helix, and it's going to very distinctly have a different secondary structure. That distinct difference is going to disrupt pretty much everything else, or probably actually trigger a repair enzyme to come through and say, hey, this doesn't fit right, I need to remove it and fix it. Right? If we try to go with cytosine, then we're going to have a separate issue. Okay? The issue with putting cytosine in there is that is going to give us the proper size gap, but would we get the proper hydrogen bonding interaction? No, and that's going to be dependent on how our cytosine's forced in there. Okay, because remember, it's attached to your sugar in this position. We can't flip that over, okay, to get the proper alignment. So if we looked at our cytosine to adenine interaction, we'd have 2 hydrogen bond donor interactions bumping up against each other, and then 2 hydrogen bond acceptor interactions bumping up against each other. So we wouldn't get a favorable hydrogen bonding interaction at all between our cytosine and our adenine. Everybody kind of see that? So what we're looking at is that hydrogen bond interaction being our favorable one, and that's the one that's going to maintain and allow for proper helix formation. One of the other features we talked about was the base stacking. So you get that pi orbital stacking. All of our nucleotides have that pi system, extended pi system. Where are those p orbitals? They're coming out at us and away from us. If we're going to look at adding another base pair over after this one, it's going to be sitting over the top of this, okay, which means we can stack those pi orbitals, okay, or those p orbitals on top of each other. That stacking interaction stabilizes the overall structure, and it actually contributes to our DNA helix stability. Okay. We'll also see a slight propeller twist, and the whole idea there was we actually optimize our pi stacking, and we minimize water's presence. Okay? Water could go through and disrupt the hydrogen bonding interactions. We don't want that in there as well. Okay? Kind of makes sense? Sure, that gives me a lot of confidence. We can move to our tertiary structure, and this is where we get our supercoiling. Okay? Supercoiling's going to become important because if we're going to try and replicate the DNA and our DNA is supercoiled, that means we've actually shrunk access to the nucleotides on the inside. Okay, so it's going to become much more difficult to actually process that DNA. So if we're going to go through a replication process, we're going to have to undo that supercoiling. Well, if it's going to make replication take that much longer, why supercoil it anyway? Why do we want to compact it? Okay, we've got a lot of information, so we do want to compact that down into a smaller space. There's another reason. We might be able to do that, particularly with our histones. Okay, when we look at a nucleus, is it just your DNA, or are there other things there? We've got other things there to allow for replication, duplicating the DNA. What if there's a mistake? What else is present? You're going to have some kind of repair mechanism. What does that repair mechanism have to do? It has to destroy the DNA. Why do we want to compact down our DNA in a supercoil? How easy is it going to be once it's compacted down for that destroyer, that repair mechanism, to get in there to actually change or destroy the DNA? It's going to be less easy, more difficult, more difficult to get in there to process that. Okay? So we have things known as nucleases that are going in there and will destroy things. So if we don't want our DNA destroyed, we're going to have to make it as nice and compact and as difficult to get access to as possible. So when we open it up and allow for replication to occur, that's good because we can now start to replicate, but at the same time, that's also going to expose the DNA to nucleases that could potentially destroy the DNA.
So the coils basically just protect it?
So the coils are part of our protection mechanism as well. Okay? And there's a couple different enzymes that are used in trying to get that either positive or negative supercoils. You're looking at your topoisomerases, which can get separated into those two classes, which we also talked about. I don't know that I can answer that question, actually. It does kind of appear that they're setting up that situation, so that could very well be what's happening. I'm going to guess it's going to have to do with the helix itself, so that if we positively, let's see, left-handed twist analogous to negative supercoil. Well, just because the image looks like that, I don't know that I would trust that.
Well, negative doesn't mostly
Yeah. Based on the way they've got the figure set up, I'd say you're probably looking at that, and I would hope that they designed it that way with that intent. So our supercoiling, and in particular eukaryotic DNA, so in prokaryotic DNA we just have this supercoiling process that just makes the DNA loop kind of curl up on itself, which is what we saw in the positive and negative supercoils. All right. When we move, sorry, I keep going back and forth. When we move to our eukaryotic DNA, we don't have circular DNA. It's in one single strand. So when we go through and start to wind either our right-handed or left-handed clockwise or supercoiling, we end up with adding extra loops within it. Those extra loops need to be stabilized as well. Okay? And that's where we can get our quaternary structure. And this is where we can bring in our histones.
Is that really good?
Okay? So the histones will sit in that loop and help stabilize that supercoil. Okay? All sorts of theories behind what exactly is happening with this. They're still doing active research and figuring out what the exact role of the histones are and deciding what roles they're playing within each of these things. So when we're looking at our eukaryotic DNA, we definitely have the histones popping up. There's a whole slew of histones to work with. I got 5 different histones. And they'll be used to make up what's referred to as the nucleosome, which is that supercoiled DNA wrapped around a grouping of histones. The histones 2 through 4 are what make up that nucleosome, that kind of grouping that stabilize that DNA. And if I remember correctly, they go in pairs. Yes. So there's 2 H2As, 2 H2Bs, 2 H3s, 2 H4s within an individual nucleosome. Okay?
So where is H1?
That's a good question. I don't think I see it there.
What does it mean it's like now outside?
So what that's meaning is that if we look within an individual nucleosome, H1 is not found within that. It's found in kind of these single-stranded sections. Okay. The exact roles behind those aren't defined in the textbook because they don't know exactly what's going on. Sorry?
I'm just confused on that.
What this purple bead is a group of your histones. Okay? That is what's making up your nucleosome. That's H2 through H4 that come in a dimeric fashion. So we have two H2As, two H2Bs, two H3s, two H4s make up a single purple bead. Okay, and that's what's stabilizing the DNA.
And then the H1.
And then the H1 is not found within the nucleosome, it's found somewhere else on the DNA strand, which isn't tagged on that structure. Okay. And as your textbook does reference as well, it is an active area of research to decide what exactly is happening. Okay? Are the nucleosomes actually stabilizing the DNA, or are they allowing for easier replication? Okay, can they help unfold the DNA in those individual locations to allow for that? Or to look at potentially gene expression, okay, or for translation into, transcription into the RNA before you can shift into the proteins. Okay? So they don't know exactly what's happening there, we just have an idea on what's going on with the structure. Okay? The whole thing, when we put it all together, that's where we refer to it as chromatin. Right, so you've got your DNA, your histones, all of that supercoiled process, that's our chromatin. Questions on that? Anything anybody wants to add? We're definitely pushing way far into the realm of biology, so you guys may have heard something more interesting than I have. Okay. Denaturing DNA. So what we could go through and do is actually spread DNA apart in a in vitro situation, meaning we take it out and put it into a test tube effectively. How can we actually separate the DNA and allow us to actually play with those strands? So to go through and do that process, we're usually adding heat. Why would we add heat? What are we breaking when we denature? We're breaking the intermolecular forces, so what we're trying to do is cause the split through the center of those two strands of DNA. So we're trying to break all the hydrogen bonding interactions. Easiest way to break those hydrogen bonding interactions, at least non-destructively, is just to add enough energy that they oscillate enough that they can come apart. Okay? We have to be a little bit careful with that process because what happens if you add too much heat? You can actually completely destroy it. Right? So we do have to be a little bit careful when we go through that heating process. So when we're looking at denaturing, we'll just add heat. We can eventually continue to add heat until we've got it separated into two strands. Okay? At that point, what if we wanted to make it come back together?
You cool it.
In theory, we could cool it. But that whole process of putting the DNA strands together was controlled by a ton of different enzymes, polymerases, and all sorts of other fun things. Okay? If we put this into the test tube and we just add heat and then cool it down real quickly, it's not necessarily going to work. So what we want to do is allow it the proper time to be able to find those favorable interactions solely on their own with no real guidance. So to allow for that, we have to cool very, very slowly so that we can find that most stable state where we've got that perfect overlap to get back to our double-stranded DNA.
It's
Where did that go? Hyperchromicity has to do with how we can tell our DNA is unraveled versus not. Okay? So, and that's what this is referring to. We can monitor that separation using ultraviolet absorption. So if we absorb or check the absorption at 260 nanometers, what happens to absorb at 260 nanometers are the bases. Okay? When we have it in a completely stacked DNA system, what ends up happening is we now have conjugated effectively all of that pi system, which means it doesn't absorb at that wavelength. What happens is we start to split the DNA. Now we've lost that base stacking ability, and now we start to see the absorption peak at 260 because it's the absorption of those nucleic acids, or nucleobases rather. Okay? So as that starts to unwind, we can see that absorption increase. And if we were putting it back together, what would happen to the absorption? It would go down. Okay? So that process of our absorption increasing is what's hyperchromicity, is what they're referring to. you're increasing the absorption, or increasing the, chrome is really color, but it's increasing the UV color, if you will. We did already talk about this when we looked at melting point, so the point at which we're going to be able to separate these. If you have a higher percentage of G and C, so guanine and cytosine, why is that going to require more heat to actually denature the DNA? Each of those interactions has 3 hydrogen bonds as opposed to 2. It's going to require a lot more energy to break those. All right. The reverse process is known as annealing, just as a random word. Just a vocab word for that. And we already talked about how it's going to happen. All we're going to do is slowly cool and allow for it to naturally find its minimal state. We've done that before. We ever completely taken something from a solid and turned it up into a solution and then slowly cooled it down so that we could then purify it? Recrystallization in organic chemistry is running effectively the exact same process. Okay? The next part of your kind of nucleic acid structure is we could look at ribonucleic acid. What's your primary difference from DNA? Or some primary differences, I guess we should list.
Uracil.
I was going to start to write, but I don't think writing's going to work. Let's see. We've got uracil. Yep, that didn't work. You've got uracil in RNA versus DNA has thymine. What else is a big difference? You're looking at single-stranded. Can accept that. What else is a big difference? What's that? It's ribose versus deoxyribose. Okay, we remove the alcohol, we have the alcohol still at our carbon number 2 in our ribose ring, okay, in our sugar. When we moved up to DNA, we removed that alcohol functionality, and that's why it's a deoxy. Okay. I don't know why I kept this figure here. Doesn't really make sense. With DNA starting up at the top, we can go through replication where we can repeat the DNA or duplicate it, and that's our replication process. We could potentially also convert it over into RNA. So that process of going from DNA to RNA is known as transcription. Once we've got the RNA strand, that can now leave the nucleus, and that can then be converted into proteins via a process known as translation. Why might we not just use DNA? Why do we convert to an RNA system? Okay, but if we look at the origins of life, one of our arguments is that RNA is the origin. Why did we create DNA in that case? DNA is potentially more stable for what reason? We got a double helix. That's a big reason for why DNA is more stable. What's another reason? The double-stranded is going to be part of your double helix. The deoxy part. We've lost that functionality on our secondary carbon. We don't have that free alcohol to potentially go out and do other chemistry. Okay? So why is it nice to have DNA as a secondary system and not directly translate into proteins? Why? We could encounter mutations in the process of that translation process or transcription process. We want to have a solid system that has a high fidelity that has very minimal errors in it. If we use DNA to both replicate and generate our proteins, we run into the issue of increased potential for mutations that could potentially lead to cell death. So what we want to do is have, I don't even know why I'm recording because I can't write anything. I don't even know if it's actually recording for that matter. And it's not advancing slides. Oh, no, I put some information on there. Our big difference, we got single-stranded. And it's actually going to act as our, really I should say translator, I guess, allow us to actually get out into the protein sequence. Okay, so that can get our information from the nucleus out into the real world where it can then be processed. Which we talked about. And then we also have our DNA dictating our RNA sequence. Okay. Is there just one type of RNA? Nope. What type of RNA do we have? Transfer RNA, which is our tRNA, rRNA, mRNA. I got 3. SI, what's SI? I got T, M, R, SI.
Yeah, there's
We got R.
Yeah.
Good cheat. Look at the next slide. Let's just do that.
Oh, SN, yeah.
So we've got a variety of different types of RNAs. We're going to separate the RNAs based on their function. So each of these different types of RNA are still ribonucleic acids, but they carry very particular functions. Because the function's different, of course, we've come up with a slightly different name for it. Some of those names actually make sense. Like tRNA, the T is for transfer. What is it transferring? What's that? I would actually argue it's not transferring our information.
tRNA, isn't it just bring in?
It's transferring your amino acids. Okay, so it's going to transport an amino acid so that we can actually start to build our protein structure. Okay, how does it know what amino acid to bring in?
It only goes for, or it only fits like the original amino acid.
So the tRNA will only bind one type of amino acid, but how does it know that it should bring that amino acid in for the proton, or for our protein structure?
What's the codon? The triplet codon.
Triplet codon that matches what?
The amino sequence. I don't know. Triplet.
RNA. There has to be something that's actually coding for it that it's actually interacting with to bring in that amino acid. So we have to have another strand of RNA that the tRNA can match. Okay, what's that strand going to be? That'll be our messenger RNA. It's the one transporting our information from the DNA out into the cytosol so that we can actually start to build our protein. So our tRNA can match with our rRNA, and in that matching process, it'll bring in an amino acid, say, hey, here's an amino acid, do something with it. Okay, how is it going to do something with it? Because the tRNA just brought it in. The mRNA just says, match me. We need something to actually start linking those amino acids next to each other, and that's where we're going to our ribosome. What's in our ribosome? All sorts of fun things. One of the things that's in it is our rRNA, or our ribosomal RNA. It's part of that translation process. Okay? So those are going to be your big ones, those big 3 is what I would argue. Those are going to be the big 3 that you're going to really be responsible for, that you're probably expected to know a lot more information about.
Variable and small.
That has to do with the size. So your tRNA is relatively small in size, and we'll talk a little bit about that size in the next few slides. Your ribosomal, you have you can have small sections and you can have very large sections of RNA. And then the same with your mRNA, depending on what you're trying to synthesize. If you have a relatively short protein structure that you're trying to synthesize, the mRNA is going to be relatively short because you don't need a whole lot of space to code for that.
So we didn't really need to worry about that.
We will still talk about these, and you're still responsible for these, but they're going to show up a little bit less frequently. You won't see people reference those quite as much as the other ones. At least that's my opinion of it. The sRNA is found primarily in the nucleus. And that's where we're getting that N from. The S, I would like to say, is small, but I'm not sure that's the case. It is actually officially for that.
Is it smaller than the T?
So we're looking at small nuclear RNA. What that RNA is going to do is process mRNA before it leaves the nucleus. Okay, so when we do that initial transcription process of our DNA to our RNA and to our mRNA, okay, there's a bunch of extra junk that gets coded along with that we don't want to show up in our amino acid sequence. So what ends up happening is that our small nuclear RNA is going to process that and say, okay, I don't want this information, and it's going to get rid of it before the mRNA actually makes it out into the cytosol so that it can be processed. Okay, so your small nuclear is just a processor on your mRNA. That process is referred to as snRNP. I forgot what that directly referred to, but it's typically said SNRPs. And it's part of that processing of cleaving out information. Your siRNA, that's still an active study, or active area of study. What we're looking at is interfering, is your I portion of that, and that's going to affect gene expression, okay, in some form or another. So what it can potentially do is increase gene expression or decrease it. Probably decrease considering the name interfering, but that could very well have just been what they first discovered. Okay? So we got to be a little bit careful with some of the names, because usually our names just come from first thing that they saw. Okay? So those are the primary types of RNA that we're going to talk about. We've got our tRNA first. It's referred to as tRNA because if we look at its secondary structure, which we got in the middle there, kind of looks like, I can't draw on that, kind of looks like a T. So there's our tRNA, okay, as well as our transfer. It's one of our smallest RNAs. We're looking at 34 to 94 residues. Or 34 to, no, I said that right. No, I didn't. 73 to 94 residues. So we're looking at that many nucleotides being involved in that sequence. Okay, the other thing that you might see reference to is something known as a Dalton, which did pop up in the lab for those of you. Not everybody's in the lab this time. What does the Dalton stand for?
It's the weight.
Yeah, what units is that weight?
Daltons.
It's grams per mole. It's the same thing as grams per mole. Okay. Why did they switch over to Daltons as opposed to grams per mole?
Biology.
That's the best answer I've got. Because Dalton was super famous, and they decided to give something, make him famous for something else. So now we can't ever forget his name. You'll typically see it referenced as a 25K or maybe a 25M. What's the K stand for? 1,000. So we're just looking at a 25,000 gram per mole molecule. Okay. It's going to carry your amino acid at the 3' end. So if we take a look at that secondary structure, you'll notice that we see some base pairing within it. So within that strand, it folds up on itself, and it does get hydrogen bonding interaction between those base pairs. So it does sort of have a helix, okay, or that base pairing system, 2 strands, but it's all intra-strandular. Okay, that makes sense, right? Okay. In that process, we're going to have a little tail hanging off on the 3' end. That little tail is where it will pick up an amino acid. Okay? So we've got that intra-hydrogen bonding. Within each of those hydrogen bonding interactions within the strand or intramolecular hydrogen bonding, we end up generating a alpha helix. So very similar to protein structure or even DNA within those systems. It will need to be activated. To get activated, what we're looking at is another enzyme. Okay, and in this case, it's tRNA enzyme. Why tRNA? Because that's what the enzyme is trying to do. It's processing tRNA. So that's where our name is coming from in that. Whoa, I did not do that. What else based on that name might we decide? Nucleotidal transferase. So we're looking at probably some nucleotide sequence and some kind of transferring property. Okay. What is that transferring property potentially doing? Very well be actually pairing the codon, our nucleotide sequence, with the proper amino acid sequence so that we could actually fuse an amino acid to the tRNA. Okay. That transferase enzyme will also go through and process, or kind of, not process, proofread? Yeah, proofread works. And on good molecules of tRNA, so if the tRNA has been generated properly, it says, okay, that's a good molecule, that's allowed to go out and do its job. To go through and verify that, yes, that is indeed a good molecule, it will add cytosine, adenine to the 3' end, I think it's 3' end. And with that little tail, it's now activated and it can do its job. If it's bad tRNA, so if there's something wrong with the overall structure, that enzyme has to also come up with some way to mark it. Okay? So shoot, what's that? There's that famous phrase about everything looks like a nail to a hammer. Okay, what's the role of this enzyme? It tacks on CCA, okay, for good stuff. What's it going to do to bad stuff? Well, if its job is to only attach CCA, how can it tag bad stuff? We'll just tag it twice. So that double tagging now says that tRNA is not good. It needs to get destroyed somewhere else and put back together later on. Okay. One other thing to kind of note, that once tRNA has been properly processed, you'll notice that it does not look like a T anymore. Okay, we pick up a general L shape. So this is showing your helices within it. It's kind of a neat picture as far as I'm concerned, as far as what that looks like. So you pick up kind of that L shape. Okay. Questions about your tRNA? Your ribosomal RNA, it is found in the ribosome. That was a ... because you're supposed to tell me that, but oh well. Whatever. It makes up roughly to 60 to 65% of the weight of your ribosome, so it's a pretty large contributing factor to the ribosome. What's the rest of that weight? Proteins. We really only have three primary characteristics within our enzyme, or within cells: DNA, RNA, and proteins. Okay, if ribosome is all outside the nucleus, it can't be DNA. And if we've now said that 60 to 65% of it is RNA, the only thing left is proteins. So proteins are going to contribute to the last portion of that. This is the X-ray crystallograph of a ribosome coming out of E. coli. You'll notice that there is some coloring patterns in this, and that's because that molecule, or our ribosomes, are made up of varying sections. Okay? So the first cleavage, as you could see on the right-hand side, more a gray bulk structure, and on the left-hand side we see this blue structure. For a ribosome to be appropriate, it actually has these two pieces. Okay? And they're typically referred to as the 30S and 50S ribosomal units. What does that S mean? Nope. It's not a bad idea, though, but no.
Size.
Did I actually write it down there? Kind of has to do with size, but not directly. One of the ways that we can actually determine experimentally what's going on with these individual molecules and that that we have these pieces is we have to somehow come up with some way to separate them. If these two pieces make up our ribosome, we're looking at something free-floating, a lot of hydrogen bonding interactions. Their solubility is going to be very, very similar. So how can we go through and separate them? This one's more massive, so it's kind of has to do with size, but it's not perfect. It's to do with density. Okay, so what we can go through and do is spin these down through an ultracentrifuge, and they will separate based on their densities. That 30S and that 50S has to do with the density of that individual piece. So the 30S is a less dense piece than our 50S. I definitely did not include what the S meant up here. That's my bad. It's named for the guy that actually discovered it. It's in your textbook. I want to say Cephidex, but that's totally not right. We can then go through and break it down further. One of the things that's interesting with this whole density separation, what is the density of the overall ribosome? 70. It's not an additive process, okay? Because it's not just mass that's changing, it's also the volume that's changing. So it turns out when we put those two ribosomal halves together, we actually end up with not a sum additive effect of your densities. Okay? Each of those sides can then be further broken down. In your 30S, you end up with a 16S rRNA, and then a whole bunch of different proteins that go along within that piece. Within the 50S, that can be broken down into a couple other sections of your two primary sections of your rRNA, a 5S and a 23S rRNA piece, along with 34 different proteins. Okay? Why might you have said 80S for our ribosome from E. coli? Additive, and what's also at the bottom? AS. What is E. coli?
It's in your.
It's prokaryote. So we have a difference between a prokaryotic and our eukaryotic ribosomes. They have slightly different sizes. Your prokaryotic is 70S and your eukaryotic is 80S. So they have a slightly different density between them. Okay? So you can't do your little math tricks in your head. Okay? Messenger RNA, that one. We're looking at our, what's that?
They're not true.
That's true. They're legitimate mathematical steps. We're looking at RNA that actually transfers the information from your DNA out into the cytosol. Okay? So it's what's coding or transferring the coded information from your DNA out for protein synthesis. It does tend to have a very short lifespan. Why might that be the case?
Because you want to keep replicating over and over again if you don't need it.
If we don't need it, we don't want it sitting out there. Because if it's sitting out there, what's likely to happen? It's going to start to get translated. So a lot of the processes that we're seeing have to do with, if it's there, it's getting used. Okay? So if we don't want that protein being made right away, we better not have that sequence of mRNA floating around. So mRNA tends to be generated when we want it. So there'll be some cell signal that says, hey, you need to start translating or transcripting that information first so that we can then translate it over into a protein. Okay, so the lifespan for your mRNA is relatively short. Okay, it is synthesized from your DNA strand starting at your 3' end. Yeah, I don't know why I actually read that off. Before it leaves the nucleus, that's where we have to get your SNRPs occurring. So your small nuclear RNA are going to come in and process that messenger RNA and remove the information that doesn't need to be there before it leaves the nucleus. Okay? So we get two sections or two pieces of information. We get good stuff that we actually want to leave the nucleus, and then we also have bad stuff that we just want to kind of get out of the way so it doesn't actually translate across. Okay? So your exons are your good stuff, your introns are your junk stuff. Okay? Very scientifically.
mRNA get processed through like the Golgi apparatus or whatever.
You just said a whole bunch of stuff about biology. For sure, I know it does get processed in the nucleus. It may get another processing in your Golgi apparatus. Any questions about your DNA or RNA? You'll notice I don't have a special slide on your sRNA or your siRNA, and that's because there really isn't a lot for me to say, other than the definitions we already gave you. Okay. That means we'd be pushing into chapter 10. So I don't know why I wrote 13. It's 10 to 12. I don't know why I'm actually attempting to write, because I can't write on top of this. It's chapters 10 to 12. 10 is referring to replication, so taking your DNA and turning it into new DNA, okay, or so that we can then do cell division and propagate that cell. Chapter 11 is about transcription, so converting your DNA into your RNA, and we will potentially look at reverse transcription as well. And then we can move from RNA into proteins, and that's our translation process, and that's chapter 12. Chapter 13 is looking at all the experimental ways that we could mess around and do this kind of stuff, which we won't talk about in the lecture, and if we have a lab on it, we'll talk about it in the lab. Okay? But those are our primary categorizations for those chapters. So we're going to take a look at replication. So just in general, what needs to happen? I will say also that these slides, at least the first few slides that we'll probably get through, I did get posted yesterday, and then I posted the rest of the chapter 10 stuff literally this morning while we were sitting in here.
You have to split it.
Okay, so we definitely need to be able to split the DNA molecule. Okay, we have to get those strands separated away from each other, but before you can split that hydrogen bonding interaction, what else are you going to have to do?
You have to find where you're going to split.
You're going to have to uncoil it. So you're going to have to uncoil the DNA, then you can split it into the strands. What else do you have to do?
Read it.
You're going to have to read it and interpret it. Once you've got it interpreted, what do you do?
Proofread.
You find it.
Well, once we've got it proofread, or sorry, not proofread. Once we've got it read, we're going to need to actually start putting DNA into it. Once DNA has been incorporated, then we can start to proofread what we've done. Okay, so we've got a whole bunch of different processes that we're going to have to step through. Okay, all of those have particular roles going on with them and different complications that go with it. So we've got the unwinding of our DNA, separating our strands, selecting the correct base pairs, adding the base pairs to the new DNA. Okay, it's not just putting in a base pair, because we also have to then form that phosphodiester linkage between each of those base pairs as we add them in there. Okay. Then we'll have to go through and fuse the backbone. We also are going to have to proofread, check for errors. Not only do we have to check for the errors, but we then also have to fix the errors as we find them. Okay? And then in theory, everything has to get wound back up. Okay? So there's all of these individual processes. Some of them happen naturally on their own. Some of them need something to help facilitate and push them along. Okay? Is there an added wrinkle when we're looking at our DNA replication? Is there a difference between prokaryotic and eukaryotic DNA?
Yeah.
Yeah, what's that difference?
One is my binary fission, and
The DNA replication process is similar, so I'm not sure what you're going with the binary fission. What's the issue between prokaryotic and eukaryotic DNA? Primary difference?
one's circular. And
Circular versus straight, or a single or
Linear.
a strand, linear. There you go, linear makes more sense. Okay. Would that have an effect on the overall translating process? Yeah, that sure. Sorry, replication process. Yeah,
Well,
that should have.
Prokaryotic runs in two directions, and
Eukaryotic runs two directions too. They
Well, when I
both go.
say two directions, I mean like in eukaryotic it does two strands at the same time.
Yeah, like DNA
In
replication.
prokaryotic it runs in two directions at the same time.
They both run in two directions.
Which one's circular and which one's linear?
Prokaryotic is circular. I can in fact, here you go. Here's the figure. You got eukaryotic on the right, and you got prokaryotic on the left.
There's a difference between like, I know there's a difference like with replication because there's like
It's not going both ways in eukaryotic though, isn't it? Like entering-
Well, if it's only going one direction, you tell me. If we go one direction, what would I expect to see over here? A new strand and a blue strand, right? Red and blue. If it's only going that one direction, what would I expect to see happening the other direction? Absolutely nothing. What's happening the other direction? It's also expanding. So I'm just going based off of what I just read and what I see in the slides. So there could very well be some kind of an issue there. There's definitely more to it than just saying that these are what's happening, but we definitely see those expanding. When we go through and synthesize DNA, do we get it completely new? So we look at it and say, oh, it's A and T, so I'm going to take a new A and T and put them together. No. What ends up happening?
You get 1A.
We take one strand and we put on a new partner with it, and then the other strand we put on a new partner with it, which ends up referring to this semi-conserved process. So our old strand ends up being pushed through the generations. Okay, but it's always getting a new partner in each case. So that's what they're trying to show within this figure. So if we start with our original strand, in the red, if we go through a single replication process, we end up with a single red strand and a single blue strand. If we then go through another replication process, what happens? Okay, we start to split it again. So we don't ever get rid of our original strand. It ends up continuing to process through. How might we have tested this and proved this in an experiment?
Tag it with
It's dealing with density. Right? We can go through and change the density of our parent strand. How could we change the density of that parent strand? I know it's a really small figure, so maybe you'd be able to cheat.
I'll use radioactive.
Radioactive isn't going to change the density, but you're right with isotopes. We change the isotope. What changes with the isotope?
Oh.
The mass, but it's still in the same volume, so what else is changing with the isotope? Density changes. Okay? So what we can go through and do is we can grow cells in a particular isotope-rich environment, which means when it starts to make those new DNA molecules, what ends up happening? It starts incorporating that heavy atom into the DNA structure, which then what happens to the DNA structure? It becomes more and more dense. Once we now have decided that, okay, we've got our super dense DNA, what can we do now? Now what we want to do is actually test our theory of, is it completely conserved? Is it not conserved? Is it destroyed? What happens to it in that replication process? So now what we can do is get rid of that dense or enriched isotope mass. So instead of using, in this case they're using nitrogen, instead of using nitrogen 15, we can shift to nitrogen 14. Why would we say nitrogen 14 and not a different one? What's the atomic weight of nitrogen? 14. The most common isotope is our nitrogen 14. We could try and use a different isotope like nitrogen 16 or 13 or 17. What's going to be the issue with that? It's much less common, which also means it is much more expensive. Okay, so we want to go as cheap as possible. So we'll enrich our system so it has a whole lot of this heavy or dense DNA. We can then shift the medium back to normal nitrogen and wait for it to replicate out. In that replication process, once the cells divide, we can take some of them out, chop them up, and actually separate the density or isolate the density and determine the difference in the densities. And that's our density gradient ultracentrifugation, which is the same technique or a similar technique that we used previously when separating out our ribosomes, if not the same one. Okay? We can then see where did the DNA fall. Okay? So we've got an initial where it's all very heavy, and we have an initial where there's no heavy nitrogen, where it's all much higher up. So what we've got in our results is that we have light DNA, heavy DNA. What happens after one replication cycle? Well, if it's conserved, we would expect heavy DNA and light DNA as our result. If it's not conserved at all, what would we expect in our result? Not conserved. If it's not conserved, what happened to the heavy stuff? It was destroyed. We would expect all light DNA. If it's semi-conserved, what would happen? You're going to have a mix of both, which means where's our density going to settle? Somewhere in between. If we then continue to let our cells replicate, we can then go through and look at another sequence or another lifespan of our DNA. And again, go through that same process and see what happens. Okay? And so what they ultimately decided is just look at all the possible relationships, what comes out of it, and then spit it out and see what your answers are. And in that spitting out process, they looked at it and noticed that what we've got is semi-conserved replication process. All right, so we don't ever get rid of our parent one. The whole replication process starts at an origin, a particular origin, which I did not see actually specified exactly what it was within your textbook, but what it did reference is that the difference between your prokaryotic and eukaryotic is in prokaryotic there is one origin. Starts at one spot, processes the whole strand out, and then we're done. When we look at eukaryotic, it processes in multiple different spots. Okay, so each of those origins can then translate out, and eventually we can copy everything through. Okay. One argument for why I really don't think the unidirectional works. If this is my origin, and I only translate this direction, what happens? Do I ever translate this section? Nope. Okay. So what you might be thinking of is how does it go through and process that? What end does it allow to start at? When we go through and process DNA, where does it go? 5' to 3'. So when we split that DNA, if we look to the right, we'll look at our 5' to 3' DNA. When we look to the left, is it that same strand? Nope, it's a different strand. So that's why we can propagate out both directions. And in each of those cases, we're going to have slightly different mechanisms depending on which direction we're running. Okay. But we're still maintaining that same directionality. It's still I think it's 5' to 3'. Okay? So everything starts with an origin of replication, and then we get that bidirectional functionality. Okay? Bidirectional does not mean we're doing both 5' and 3', 5' to 3' and 3' to 5' as a primary function. What it means is that we're going both directions to replicate our DNA. Okay? And depending on which direction we're going to have a slightly different process or a slightly different strand we're analyzing. Okay? So our processing of this is where it definitely changes because I switched up the order from the slides I had initially posted. The first thing we're going to have to go through and do is undo the coiling. We have to get that so we can get access at our DNA. So we can look at our DNA gyrase, which is our class 2 topoisomerase. And what that can do is induce or reverse our supercoiling. We're going to want to reverse it so that we can get access to the nucleotides. Okay? Once we've gotten that reverse process, we can then also start to unravel our double helix. So our DNA gyrase is going to get us away from that supercoil issue or relieve supercoil issues as helicase undoes your DNA helix. Okay? Why is it undoing the helix? What's the name? Kind of took it from the name. We're looking at it just trying to destabilize that so we can get access to it. Okay? At that point, once it's now split, we've got kind of an issue. Okay? Because what we've got is single-stranded DNA. What's the issue with single-stranded DNA? It could go back together, which ultimately isn't that big of a deal, but that's a problem. It's now exposed to being able to react with something else. That something else may not be what we want it to react with. So we need to come up with some way to protect the DNA once it's actually opened up into those single-stranded situations. That's where we can bring in our single-stranded binding proteins. And that's going to stabilize your single-stranded regions so that it doesn't get destroyed by, say, nucleases that are floating around trying to destroy bad DNA floating out there.
Well, there's also the string. When you open up, you can't, it'll like put a strain on the DNA.
As you unravel it, yeah, what's going to happen to the coil? You're going to start inducing supercoiling on the other end. How can you minimize that supercoiling? Your DNA gyrase is going to do that for you. Your singles, the SSB, because I already forgot the thing, is looking at just the single strands to kind of keep them protected and not being folded back on or forming back into the double-stranded DNA. So what you're referring to is absolutely true, but our gyrase is taking care of that, not the single-stranded binding protein. I didn't think of the second term on it.
I was just wondering why opening it up makes it supercoil up.
So if you take, couldn't use it anyway, right? If you take a string, you notice the strings are all kind of wound together. If you pull, those separate, you'll notice that it starts to get tightened, and eventually you can't pull anymore, it stops, and that's because it's so supercoiled. DNA is going to run into the same issue. So what you need to do is actually relieve that stress. Relieving that stress is going to happen with your DNA gyrase. That makes sense. And then as Stephanie kept suggesting a couple different times, to start off your DNA synthesis, we're going to add in an initial chunk of nucleotides. That initial chunk is what's going to allow us to then continue to propagate out the rest of our nucleotide sequence. That's your primer. The primer is a piece of RNA, and that's put in initially with your primase. Okay? That primer gets put in so that we can then start to add the nucleotides on in sequence in the 5' to 3' direction. Okay? What about the 3' to 5' direction? That's where we're going to have to come up with a different mechanism because everything's following this 5' to 3' issue. If we're going to try and protect that other sequence of DNA, we're going to have to slide something in to put those nucleotides in as well. And what ended up happening in that case is we're bringing in other sections of fragments, and those are our Okazaki fragments, which for some reason I don't think I've got in the slides. So in our normal direction, our replication starts with your primer. Okay, so from your primer, what we're looking at for our growth is that we've got the OH on our 3' carbon that can act as a nucleophile that will attack then the phosphate of our next nucleotide to generate our phosphodiester backbone. Okay. You'll notice that we aren't looking at a monophosphate nucleotide. We're looking at a triphosphate nucleotide. Why would it be a triphosphate?
Energy.
It's energy. That's all it really is. We're using a triphosphate because that is a higher energy state. If we have the oxygen come in and attack, what can we kick out? A really good leaving group. If we didn't have that phosphate there, what's our really good leaving group? If anything, it might very well be our ribose ring, which does not the right thing. So we need to be able to have a leaving group to kick out, and that's why we're going to use that triphosphate system. Okay? So it's going to increase the energy of our reactant to provide enough energy to actually form that bond fusion. So when we're looking at our direction, we're growing from the 5' to 3'. I definitely don't like that whole phrasing system, but as long as people understand what that means, I'm fine with that. When we're growing from our 5' to our 3', it's our 3' oxygen that's attacking the 5' of the next species. Okay? Which, if we look at the initial start of our 3', where did it start? At our 5' end, and that's where they're saying the 5' to 3', 5' to 3', 5' to 3'. As long as that makes sense to you, I'm fine with that. A little confusing to me. Okay? Questions on our replication slide here? Okay. The catalyst of your phosphodiester bond is happening through your DNA polymerase. Okay? So your DNA is synthesized from your 5' to 3', okay, towards the unzipping of the DNA. Why would we catalyze in that direction? Why not catalyze the reverse direction? What happens as we unzip? What do we expose? Single-stranded DNA. What do we want to do to that single-stranded DNA? We want to get that protected and not destroyed by nucleases as fast as possible. So as soon as we unzip, we want to put in a nucleotide so that we can prevent it from getting destroyed. Okay? So we're always going to be synthesizing towards that unzipping process. Okay? So the 5' to 3' works pretty well as far as that. You'll notice, though, that as you unzip, we're also exposing that bottom section. The enzyme doesn't catalyze the 3' to 5' end. Good, I do have the Okazaki fragments on here. Okay, so we need something else to account for that bond formation. Because that strand is now forming in a slightly different mechanism, we can then refer to the two strands by two different names. The one that's forming as kind of a standard, adding nucleotide by nucleotide, we have the leading strand, and the one that's not leading is falling behind, so it's lagging. So we've got our lagging strand. So we're looking at two kind of different mechanisms between how we incorporate the nucleotides for each of those strands. Okay? So if we look at the synthesis for the other strand, okay, we still need to maintain that 5' to 3', but we don't have the ability to form those bonds as easily, okay, because we'd end up exposing the 3' end first. That's where our Okazaki fragments can come in. We end up putting in sections of DNA single strands that then match, and then we have another enzyme come through and start to link those fragments with each other. Okay? That linking process is going to be done with our ligase. Okay, the DNA ligase is going to help complete that down. Okay? How many nucleotides are in an Okazaki fragment? I forgot to write down the number.
Looks like it's different.
It's actually a pretty insanely high number from what I remember from the textbook. Somewhere in the thousands of nucleotides. Yeah, holy shit is right. Okay. But I mean, anything more than like 6 is pretty impressive to me, because how do you already have that DNA section already set and ready to go, right, to fill that gap? So that's kind of a neat process. Your DNA polymerase needs the following things to function. Right? So you're going to need all of the deoxyribonucleotides. Why would you need those? When you're synthesizing DNA, what do you think you're going to encounter? All of the nucleotides. So if you don't have all of those nucleotides, what's going to happen with the polymerase? and it's going to stop functioning as soon as it hits a spot where it needs that nucleotide. Okay? Or potentially incorrectly incorporate something. Oh, crap. Definitely out of time. The other things we'll need, magnesium ion and your RNA primer to get it started to allow for that process to go. There's several different types of DNA polymerase. We'll pick that up on Tuesday, and we'll also play our little game as well.